Hello friends, is Botany. Next episode. In this episode, we'll be discussing principles of inheritance and variation, seventh one. One more we need to finish this. Anyway, we can proceed. So before it, I will share the screen so it will be convenient. I think screen came. So we can proceed. So what all things we'll be discussing in the seventh episode of principles of inheritance and variation? Basically, three topics we'll be doing. That is sex-linked traits, under which three types: mutation, a big one, mutagens, small one, and questions, 5 theory, 5 neat model with the key. So we can proceed. First is sex-linked traits. So when we say sex-linked traits, it is normally connected with the X chromosome because X is present in both the male and female in humans. Sex linkage discovered by Morgan in connection with Eichler and Drosophila we already studied. All sex-linked characters with X normally show crisscross inheritance. Remember, X as well as Y chromosomes are sex chromosome, but mainly X is present in both the male and female. Three different traits can be seen under this heading. One by one, we can proceed. First is Hollandric traits. Hollandric traits itself means it is Y-linked. And it is basically linked with the Y chromosome only. So it means from male to male only, character will be inherited because Y determines maleness in humans. That is why a famous statement you will be remembering, only males can produce males. Thus, Hollandric traits only seen in males, Y-linked. Reason is Y-linked. And some of the characters are typical traits. One is hypertrichosis, that is growth of hairs, a little bit more hairs on pin of the ear. Then TDF, testis determining factor. There is Porcupian skin. I think that idea is clear. Now we can proceed to sex influence traits. Sex influence traits, which are influenced by the sex of the bearer. If a male has one recessive allele, then it will appear. But it will take two recessive for the female to show the same trait. It is also suggested these traits appear more frequently in one sex than the other. When we say in this manner, it may not be clear, but when we observe the example, it will be clear. Typical example is pattern baldness. It is popularly seen in males, connected with the testosterone. Second is short index finger. See here. This will be smaller than this one, ring. Short index finger. So these two are sex influence traits. Now comes third one, sex limited trait. So the term indicates limiting to a sex. These traits are again autosomal, can be seen in both sexes, but will be expressed. There comes the term in one sex only. Here also sex hormone play a role like previous case. Typical example is two photographs are given. One is beard in man. Second is brilliant plumage in peacock. Third is milk glands or mammary glands in females. I think these three features clear with example. Popular question is example, order one, etc. NCERT, this much detail not given, but it is a popular question. That is why I explained it. Now we can proceed to next page. A big one. Next item is called mutation. So we can start from the base. The mutation came from a Latin word. It is mutationum, a changing. That is the meaning. The mutation was given by Hugo de Vries, photograph given here. Worked in evening primrose, that is given here. Evening primrose is scientifically called Enothera bainis, family Onagraceae, dicot, and you'll be remembering sexual reproduction flowering plants. It is having diploid endosperm, abnormal behavior. Normal angiosperm, endosperm is triploid. Here it is diploid. That item I discussed in sexual reproduction flowering plants, why it is diploid. Now, famous mutation theory of evolution was also proposed by eukaryotes. And what is the definition for mutation? Sudden, drastic, heritable changes are called mutation. And in general, when we say mutation, it is deleterious, no use, waste, adverse effect may be there. But in plants, some mutations are useful. And remember, mutations are a basic tool for evolution. And this artificial mutation, etc., we can apply in, utilized in agriculture, industries, medical aspect. Reason is DNA changes can take place. Now, four basic terms: mutagen, mitogen, pollutant, and carcinogen. So what is mutagen? Mutagen, substances that can cause mutation. Gen means production. Mitogen, substances that can cause mitosis. Pollutant, substances that can cause pollution. Then you can observe how much pollutant it is. Then carcinogen, substances that can cause cancer. Now, different classifications are available for mutation. General purpose, we can divide mutation into three types: genomatic mutation, chromosomal aberrations, gene mutation. Remember, NCERT, very little items are given, but still little bit more we have to extend or expand. So first is genomatic mutation. It is concerned with the number of chromosomes, and it can result in clear effects into the phenotype and can be classified into aneuploidy and euploidy. Mutation, I told you, big area, different classification, nomenclature, etc. available. Popular one, we are proceeding. Heteroploidy. Hetero means different. Ploidy generally means chromosome number, that way. So heteroploidy means phenomenon of variation in the number of chromosomes. That is heteroploidy. Because aneuploid, etc. will be coming under it, will be proceeding. So next one is under genomatic mutation. First group called aneuploidy. What is meant by the term aneuploidy? See, it is a situation of having lesser or extra chromosome than the normal diploid chromosome, complement of the organism. Either lesser or extra than the normal diploid chromosome complement of an organism. Two types are identified under aneuploidy. Lesser means hypoploidy. More means hyperploid. Hyper means always more. Hypo means always less. Organism having any of this, they are called aneuploids. And in such organism, suffix of the name is somic, S-O-M-I-C. Aneuploid normally arise due to an important question, non-disjunction. Here everything mentioned, non-disjunction of the chromosome in homologous pair, causing extra chromosome in one gamete, and the other gamete will be deficient of one chromosome. If fusion takes place between these two, or with even normal gamete, different aneuploids are resulted. Basically, four types will be formed, that is under two division: hypoploidy, monosomic, that is 2n minus 1, diploid number minus 1 chromosome; nullisomic, 2n minus 2. Now hyper, more, trisomic, 2n plus 1, tetrasomic, 2n plus 2. The popular question, this number. Some question I put in the question session. Typical example, in human beings, monosomic will be, trisomic will be this way. So monosomic will be 2n minus 1. So 2n is 46 minus 1, 45. I think idea clear to all of you, important item. Now aneuploids. Aneuploid means difference, either hypo or hyper. That is why it is always called hetero. Examples: Klinefelter syndrome, Down syndrome, trisomy, 2n plus 1, that we'll be studying in next episode. Turner syndrome, it will be less, monosomy, 2n minus 1, so it will be 45. And this is a typical diagram for it. Here total 2n 8. Here one extra got non-disjunction, that become n plus 1. So normally n should be 4, 4 plus 1, 5. Comparatively here, 1 will be less than the 4, that is n minus 1, 4 minus 1, 3. I think this is clear to all of you. Now comes the term euploidy. This is the condition in which an organism either loses a whole set of chromosomes or acquires one or more additional sets of chromosome over and above the diploid complement of two sets. I'll explain it when it comes down, it will be clear. Now, classical definition, euploid means an organism will lose a set of chromosome or will have one or more extra set of chromosome over diploid condition. Under euploid, two headings will be there. One is called haploid, other is called polyploid. Haploids, often called monoploids. That means monoploids and haploids are identical in diploid organism only is an important item. So monoploid and haploids are the same in diploid organism, while they are different in tetraploid or hexaploid organism. Diploid organism, haploid and monoploids are the same, but when we come to tetraploid or hexaploid, higher ploidy level, it differs. Now haploid. Haploid is N, already you know. So haploids have only one set of chromosomes. Since haploid have only one set of chromosome, it can be used for mutation experiment because character will be immediately expressed. Generally, haploids will be smaller in size. I think idea clear. Now we can proceed to next page. Polyploid. Poly means many. How it differs from previous one. Polyploid means increase in haploid set of chromosome like normal diploid. Imagine it is 12. So polyploid haploid sets will be added. Haploid is N. So here you can find 2n plus n, 3n, 3n plus n, 4n, 5n, 6n, that way it proceeds. Main reason for polyploid is the failure of cytokinesis. Colchicine will be remembering can induce polyploidy due to preventing spindle fiber formation. Cell cycle question. And this colchicine is often called mitotic poison. Plant you will be remembering Colchicum autumnale. Family you have to learn it. So polyploidy is more in plants in comparison with the animals. Generally, polyploids in animals will be sterile. Now, polyploids can be classified into two. Autopolyploid and allopolyploid. Autopolyploid basis only we need. AA will be diploid. So autopolyploid means one more A will be added, so it becomes autotriploid. One more set will be added, so it becomes autotetraploid. 1, 2, 3, 4. 1, 2, 3, that way. Same type genome will be increased. While allopolyploid, allo means different, allogamy. If you cross AA with the BB, F1 will be capital A, capital B. That way you take. And it is 2n. It is the chromosome is doubled by using colchicine. And we'll be getting capital A, capital A, capital B, which is otherwise called allopolyploids, otherwise called amphidiploids. That is doubling of chromosome after getting F1, after hybridization. Now comes chromosomal aberrations. So what is meant by the term chromosomal aberrations? See, aberration means everything written, this will be easy for you. Aberration means a departure from the normal. That is aberration. And chromosomal aberration means structural changes in chromosome. So we know that DNA is the base. DNA connected with the genes. Here changes in the arrangement and number of genes in chromosome. It can be within chromosome, then it is called intra. It is different chromosome, it is called inter. So these changes can cause aberrations. These changes can be commonly seen in a common question, cancer cell. So chromosomal aberrations common in cancer cell. The four main types, important one, deletion, duplication, translocation, inversion. Now deletion. Deletion is sometimes also called deficiency, different nomenclature, different types, different classification used by different scientists. And it normally occurs when a part of chromosome is lost. Two common types, one will be terminal deletion, other will be interstitial or intercalary deletion. So from the diagram you can get what is terminal deletion here. That will be lost. So normally when deletion takes from the terminal side, only one break is needed. This is interstitial, in between or intercalary. Here what happens is PQR is the ST lost. PQR ST lost. UW is there. So break should take place here and here. So normally two breaks are needed for deletion. Diagram very clear. Usual theory entrance question. Hoping that is clear. Now we can proceed to next aberration. Next aberration is called duplication. Duplication means an extra segment of chromosome will be attached, duplicate. A result in a gene or set of genes will be represented twice in the same chromosome. Idea is simple. When you look at the diagram, it will be clear. Here you can find PQR STU VW. Here PQRS, again RS, TUVW. So RS will be extra. Abnormal block of gene is called repeat. Now, in this case, when this duplication takes place, there is natural adjustment that is not in syllabus still. A loop or buckle formation can be seen at duplicated area if synapsis takes place between normal and duplicated chromosome. It's an adjustment by nature, and different types of duplications are available. Typical example is Drosophila we can find normal I, bar I, double bar I. It is due to duplication. Not much advantage. It is a mutation. Now, duplication lead to a famous event called genetic redundancy. Frequently comes for exam, given as an option. Genetic redundancy means or may lead into evolution. And the meaning is two genes may do same function. That is genetic redundancy connected with the duplication. Now comes the last one in that sequence, inversion. Word inversion means turn into. Inversion we are familiar. Here, rotation of chromosome takes place in a linear manner at 180 degrees takes place. Inversion is basically two types, again important, paracentric and pericentric. Again, diagrams are clear. See, in paracentric, first arrangement PQRS, TUVW. Here PQSR, RS, SR. So before the centromere, it takes place. So not involving centromere, paracentric. But here, PQRS, TUVW, PQRT, S is here. So centromere both sides. So it is called pericentric, inversion involving centromere. Paracentric and pericentric, important one for exam. I think idea clear. We can proceed to next page. Next is last one. This is the last one. Translocation. So location means place, trans means shift. So translocation means shift the place, just like transfer. Shifting position. And here, translocation means a part of chromosome is shifted to another non-homologous chromosome. That is why translocation is also called an important word, illegitimate crossing over. Illegitimate. Many types of translocations are available. One famous type is called reciprocal translocation, which otherwise is called segmental interchange. That is, a part of one chromosome shifting to other chromosome. From other chromosome, a part is shifted to the first one. That way, reciprocal. Two famous examples for translocation is one, Burkitt's lymphoma, otherwise called immune cancer. Short form is BL. And it is concerned with the chromosome numbers 8 and 14, interchange between 8 and 14. Second is more famous for exam point of view, chronic myeloid leukemia, CML, cancer, both are cancer. And Burkitt's lymphoma you can find here, somewhat here. A swelling you can find here, see. Now, chronic myeloid leukemia, it is between chromosome number 9 and 22. Chromosome number 22 is called Philadelphia chromosome. That name came due to the place concerned with the work of it. It is also called PH chromosome. Now comes gene mutation. Gene mutation means changes in gene due to change in nucleotide. If the mutation takes place due to change in a single base pair of DNA, it is called point mutation, important. If the mutation is due to change in more than one base pair or even entire gene, then it is called gross mutation. And most of this type of mutation arise due to DNA replication errors, that is otherwise called copy error mutation. Again, here also many classifications available, many. First is frame shift mutation, second is substitution mutation. So what is frame shift? Frame change, entire frame. So entire frame or sequence will be changed. It is due to by the loss or addition of a single base or a segment of DNA. And that is due to insertion or addition. And another phenomenon is deletion. When you look onto this representation, it will be clear. First is insertion or addition. TTA, GCC, TH, that is normal. But A is added here after T. So it becomes TAT, AGC, CTAG. Same as deletion. TTA. So A is deleted. So TTG, CCT, AG. I think idea clear. Now, substitution. Substitution mutation can be two types. One is transition. Transition means purine replaced by purine or pyrimidine replaced by pyrimidine. Both are important. All these are important. Theory and runs. Transversion. Purine replaced by pyrimidines or pyrimidine replaced by purine. I think idea clear. Very predominant. That is why a little bit expand manner given there. Now comes mutagens. What is meant by mutagen? Substances that can cause mutation. It is called mutagen. It may be due to physical, chemical, and even biological reason connected with the viruses. Now, physical mutagen. Here only limited expansion needed because too much items you have to discuss not in NCERT. When we discuss with the physical mutagen, typical example is radiations, natural occurring radiations. Scientist is in this area, H.J. Muller, photograph I given here. Radiation used was X-rays, and the organism utilized were Drosophila. And this branch is called actinobiology. And father of actinobiology is H.J. Muller. Now, chemical mutagen. These are more dangerous. First chemical mutagen used was mustard gas. Its molecular formula given here, even though not referring to us still. By Orbach et al. Connected with the World War II. For mutagen, different classifications are available, different data, different detailed study, etc. Everything available, but that all are not at all mentioned in NCERT. Some basic aspects, little bit extra also given, that's all. After that, usual 5 theory question, then 5 neat model question with the key. And we will be require one more episode normally because other mutation we have to study. Again, important for exam point of view. So what all things we learned here? Well, just to mention. So things mainly we discussed in this episode, that is sex-linked traits 3, hypochondry, sex influence, then sex limited, then a big area mutation, who discovered it, then which is the plan, then mutation theory, then advantage of mutation, mutagen, mitogen, pollutant, carcinogen, then different classification of mutation, genomatic, chromosomal, gene mutation. Genomatic means based on number of chromosome. Then chromosomal aberration means structural changes of chromosome. Then last is gene mutation means it is changes in gene due to change in nucleotide. All mutations important. What is given there? All are important. Exam point of view, theory and runs, I told it is little bit important. That is why I thought it is better to give us an episode. Then only maximum we can share. Then mutagens, then physical mutagen, X-rays, then scientist, organism, then branch is called, then chemical mutagen, mustard gas, Orbach and al. Then different classifications that are not needed. After that, 5 theory question, 5 neat model question with the key. Here it is key, hoping that is clear. So if you like this episode, subscribe or like it, share it, comment on it. We can meet in the next episode. Most probably by this month end, I will start doing questions also, live discussion, 5 or 10 neat model question from first year and second year, preferably from November starting onwards. Okay, thank you. We can meet later.